స్టార్ట్ మా స్టార్ట్ చేయండి.
జన విజ్ఞాన వేదిక మెడికల్ మల్కాజ్గిరి ఈరోజు మనము నేషనల్ సైన్స్ డే సందర్భంగా జరుపుకుంటున్న MCMB
బురాన్ గారు మేడంది లైవ్ ఇవ్వండి. బురాన్ గారు మేడంది ఇవ్వండి.
ఎన్ని వాయిస్ వస్తుందా?
రాలేదు, మేడంది ఇచ్చేయండి. ఇక్కడ
ఓకే మేడం, విల్ యూ స్టార్ట్ నౌ?
Yes, I can start.
Okay, ma'am.
I just
Please
want
carry
to check whether my slides are visible. Yeah. Can I start now? Can I start now?
Okay, ma'am.
Yes, ma'am. Start.
Yeah. Good evening, one and all. It's indeed a very happy occasion for me to share the little science that I have been able to understand with all of you. And it's really an opportune moment because we are commemorating the National Science Day. So let me, in the beginning itself, tell you that I'm not going to talk like a doctor where we look at diagnosis of cancer or treatment of cancer. But I'll talk from the scientific end as to why one gets cancers or humans get cancers, and a little bit of the knowledge that we have gained through 50-60 years of work on how cancer develops. Okay, so cancer pertinent to the human beings is because we are all multicellular organisms. Multicellular organisms because all of us start with one cell, and in the womb we grow up to be an infant, newborn baby, and by this time this single cell has divided and become several billions of cells which have different kinds of functions. So there is order, there is size, and there is also shape during development of an organism, and it is relevant for the human as well as any other multicellular organisms. So what is the basis of growth and development? This is very important to understand before we can discuss why and how one develops cancer. So what is the basis? Cell division is a basis of growth from a single cell which divides many billions of times to form size and shape of an infant. Now, if these cells are dividing exactly as a mother cell, and each cell that is divided has given rise to daughter cells which look like the mother cell, then by the end of multiple divisions, you will just have a sack of cells. There will be no form or shape. They will just be number. So how is it that we are developing form and shape and specialized functions? The cells in our brain are different from the cells in our muscle. The cells in our muscle are different from the cells in our skin. So how does this come about? This is a very interesting question in development. So what we know today is every cell in our body knows what it should do next. That means, should it divide? Should it get differentiated and become a nerve cell or a muscle cell or a skin cell or a bone cell? Or should it sometimes even die? Okay, so how does this cell divide? So this decision comes from the external environment of the cell. It can be many things, like there can be growth factors, which are chemicals that circulate in our bloodstream. It can be forces, because there are adjoining cells and tissues. It can be sticky structures that are adjacent to the cell, etc. And based on these signals, the cell is deciding whether it has to divide, achieve specialized functions, that means differentiation, undergo cell death, or sometimes even stay at rest without dividing and wait for some time when it is necessary to divide. That means, especially when you have an injury, the cells recognize the injury and grow and divide to heal our bodies. Right? All of us have gone through injuries, especially if there is a cut on the skin, etc. Okay, so this is something that is fundamental to how our bodies develop cell division in a controlled fashion. Okay, and this cell division and what the cell has to do, that means what is the fate of the cell, is decided by signaling mechanisms. So how does a cell receive a signal? Usually the signal is a growth factor. There are receptors on the cell membrane which pass on the signal through multiple molecules, and then the cell decides whether it has to divide, whether it has to move, it has to change shape, it has to secrete substances, or it has to attain specialized functions, or even die. So there are billions of cells in our body that actually die to replace the newly born cells. And this happens mostly in tissues like the skin, the intestinal walls, where there are continuous cell division. But in some tissues, the cells are hardly dividing. Not only some tissues, many tissues. For example, in the brain, in our skeletal muscles, in our heart, those cells are not dividing. They have achieved specialized function and they're carrying out that function. So when this kind of property is called differentiation, these differentiated cells cannot divide. Okay, so that's why repairing the heart muscle is very difficult. Okay, so most tissues, the cells are in a quiescent, what we call is a quiescent or a resting stage, and they are called upon to divide only when there is a signal to divide. For example, if a liver is cut in human beings, the liver can grow back and attain its normal size. So this is the background of how signals determine what the cell has to do next. So what is it about cell division? How does a cell divide? How does it know when it has to divide? So when there is a signal from outside, then the cell enters something that we call as a cell cycle. That means it has entered a stage when it is determined to divide. So for it to divide, what has to happen first? All its genetic material and the other components in the cell have to multiply. It has to double up, no? If it has to give rise to two daughter cells. So the DNA has to divide very precisely. So how much DNA is there in our body? There are 6 billion base pairs. That means we have In the human body, there are 46 chromosomes, and in those chromosomes there are 6 billion nucleotides. These are molecules that make up the DNA. Now, if all of these molecules have to divide, that has to happen with great precision. At every time, every cell in our body has to divide. Especially when an infant is growing, there is continuous cell division. Also in the womb. In an adult, only some tissues divide, some tissue cells divide, some do not divide. Okay, so this DNA replication has to happen with precision, and sometimes because it is such a large number, there is prone to be error. But this error, nature has made that we can correct the errors if the error is detected. So very rarely you have a situation where there is a defect that is passed on to the daughter cells. So how is this error detected? What does that mean? That means in every time a cell is dividing, there are molecules that are called checkpoints. That means they check whether is all the DNA intact, has it correctly replicated. Suppose it is not intact, then the division of the cell, that means mitosis, you would have all heard of the term mitosis is prevented. Like that, is all the DNA replicated? All these 6 billion base pairs, have they replicated? If they have not replicated, again mitosis is halted. Okay, now once the DNA is replicated, the chromosomes have to divide. This is a textbook image. You can imagine a cell in division. You have two sets of chromosomes that are pulled apart. Okay, so if all the chromosomes are not aligned properly, there is two of one type of chromosome should not go into one cell, and the other cell should not receive none. Okay, so then the division is prevented. So these are called checkpoints. This is a term which is very important because these checkpoints are our guardians. If we didn't have these checkpoints, all of us, because of the number of cells our body has, number of times that cells have to divide, we would have all developed cancer because of the mistakes. So then automatically you will come to the question, how are the cells knowing when to stop? For example, if you have a wound on your skin, what happens? The cells at the edge of the wound have recognized that they have to divide and close that wound. How will they know whether to close that wound? For example, here is a wound. So there is a cut here. This cell is not meeting this cell. So this cell knows it has to divide, and it will divide from this side. This cell will divide from this side. Finally, there are molecules on the cells which, when there is an adjacent cell, it will tell the cell, stop dividing. Don't divide anymore. You would have seen people with some skinny structures coming above the skin. For us, the skin will heal normally. But in some people, the cells don't stop, and you will have some kind of, these are called keloids. Okay, they are extra growth. That means those cells have not understood that they have to stop. Okay, so there are mechanisms that tell the cell how to stop. And another way or another situation where the cells have to stop dividing is when the DNA is damaged. This is just an example. So I told you there are guardian molecules. So what happens when there is a damage to the DNA? It is recognized by guardian molecules. It will tell the stop, don't divide. Okay? It will make sure that the DNA is repaired. Suppose it cannot repair the DNA, that cell will be signaled that you undergo suicide, undergo cell death. So even though there is lot of proliferation, even though there is lot of damage, we are all protected, because in the interest of the whole organism, any cell which is damaged, or which is having a virus, or which is having defective metabolized or chemicals, the body is asking that cell, okay, you die, so that other cells are protected and the body is intact. Okay, so now coming to why we have cancer. Cancer cells do not listen to stop signals. So when there is a damage or when there is a virus, this cell is not stopping. That means there is some defect in the molecules of the guarding system. So I will describe that in the future slides. Okay. So it is like, this cell has pressed the accelerator and it is going on dividing, but the brakes have failed. Okay. Cancer happens only when the brakes are failed. Okay. And which cancer is dangerous, I will follow up in the next slides. Okay. So what happens when these guardians are lost? These are simple pictures of how normal cells look. I told you, normal cells, when they come close to each other, will stop dividing. See, they have formed a single layer. But when this signal to stop is defective, the cells will start growing like this. It will grow in a third dimension, and that is one of the hallmarks of cancer. Otherwise, it has changed shape, see, and then it is going in a haphazard fashion. Okay, the cells are not able to come close to each other. So these are the hallmarks of cancer. So why does cancer happen? We will imagine that it is because the cells are multiplying very fast. That is true. But our body has a guardian. If a cell is multiplying fast, the guardian tells it to stop dividing or to undergo cell death. So basically, what cell death, the scientific term for cell death is called apoptosis. Like, a dry leaf has fallen down. That is called, they have given a similar name. So basically, cancer happens because the cell which is defective is lost the control signal to undergo cell death rather than excessive proliferation. So this is one fundamental theme that we should all remember with respect to cancer. That is one thing. And these cancer cells, like I told you, when they change shape like this, when the cells are normal like this, they cannot move from their place. When they have changed shape like this, they can start entering the bloodstream and move from one place to another. That is pretty dangerous. Okay. So, and mind you, when we say cancer, is it one kind of disease? Certainly not. Cancer is a large group of disorders and it happens in multiple stages. Actually, it doesn't happen all of a sudden. It is just that we don't know. We diagnose it all of a sudden, but it has been happening over a small period of time because of multiple changes. Now, you might ask, is cancer a very recent disease that only present day population are facing? Did our ancestors have cancer? Certainly, all multicellular organisms are prone to cancer because any body where the cells are dividing continuously and there are defective guardian mechanisms can. How do we know that our ancestors had cancer? Many fossils of humans also could be detected with cancers. Okay, so cancer is quite a common phenomenon among all classes of animals. To some extent, in some classes it is more, in some classes it is less. So now it is very prevalent, especially in humans. Almost the incidence or the relative number of people who die because of cancer has increased because we have been able to control infectious diseases to some extent. And now many environmental factors are also adding to the increased incidence of cancer. So there are cancers, as I told you, are not a single disease. Okay, but a whole class of diseases. Cancer can happen in any kind of tissue, and generally it is classified if you have it in your skin cells or in your epithelial cells, what we call the skin cells, it is called a carcinoma. If it happens in your muscles or your bone tissue, it is called a sarcoma. And if it happens in your blood cells in the bone marrow, it's called myeloma. And if it happens in our white blood cells, it is called leukemia, etc., etc. And each of these has certain common features and certain uncommon features depending on the person who is having, depending on the patient or the individual. So we have to discuss a little bit about why a cell has become cancerous. Why is it not listening to signals properly? What is abnormal? So, many features define a cancer cell. One thing is, it is continuously having a feature as if there is a continued growth signal. Normally, cells will not always be told, okay, continuously multiply. When it has to stop, it has to stop. But in this case, it is being told, it is as if it has lost its break and it is in a continuous mode of division. And then it does not have the break, so it is evading growth suppressors. Then another feature of cancer cells is they're very smart. Normally, cancer cell is treated as a defective cell in the body, but who detects that? Our immune system. But the cancer cell has mechanisms to escape that detection, and then it now has become something like immortal, like as if it has taken amrut or something like that. Okay, so that normally our cells cannot divide forever. There, every cell has its own lifetime. Any cell you take out of your body, if you want to grow it, or within your body also, it has a specific lifetime. But a cancer cell does not have such a lifetime. And another property I told you it develops is, it can move from one place to another, and then it attracts blood vessels, and because it needs nutrition to grow. Okay, so these are some of the important properties of cancer cells. Now we have to understand this, and last 50 years of research has enabled us to understand what are the properties, so that we have a better means of treating cancer. So I mentioned that it's a multi-step process. Initially, there is a small genetic change in one of the genes. But even if that is there and the cell is asked to proliferate, it will start proliferating. Our protective mechanisms will ask it to stop. This proliferation is called hyperplasia. This is not cancer. Though it can proliferate, it is staying in the same place. But then after that, again some genetic changes take place in one or two of the cells that allow it to increase and grow very rapidly. So then this becomes a cancer in situ. These are called benign cancers or non-metastatic cancer. That means these cells do not now have the ability to move from one. Now again, if there is a mutation, they develop the ability to move from this place through the blood vessel and go to other places. This feature is called metastasis. And in the case of human cancers, or what we call as malignancy, this is a most important or dangerous aspect property of the cell. That means metastasis. Okay, if a woman has developed a cancer in the breast, as long as it is restricted to the breast, she is still safe. It can be surgically removed and nothing, the whole body will be safe. But if it has started moving through the blood circulation, it can go to the brain, it can go to the lungs, then it is very, very difficult to control. Okay, so there is a property that any reaches this stage, it is avoiding our immune system. It has these special properties and run away from the place where it has originated. Okay, that, and in human cancers, mostly death is because of this property of moving from one place into another place. So briefly, as a scientist, I cannot refrain from telling you, how do we understand that a cell has become cancerous? How do we know which is a normal cell and which is a cancer cell? So I will briefly tell you these features. One is that it can grow in the absence of signals. These signals are growth factor. Normal cells require a growth factor to grow and divide. But these cancer cells, the accelerator is jammed. It doesn't require the accelerator to be released and pressed again. Okay? So it is continuously growing. Then it also ignores the signals that tell the cell to stop, divide, or die. Okay? So that is very important for cancer cells. And it hides from the immune system. And slowly what happens, there are many changes in its DNA that make it, that it enable it to attain the property of attracting blood vessels, moving from one place to another. So these are called chromosomal aberrations. Okay. Now, how do we identify these properties in the lab? There are some simple experiments we can do. For example, normal cells, when we want to grow them, we have to provide continuously growth factors. But in this case, growth factors are not needed. So if a cell can grow in the absence of a growth factor, that means it is a transformed cell. It is almost having the properties of a cancerous cell. And like I told you, now a normal cell, when it comes next to each other, it will stop dividing. So this cancer cell does not have that stop signals. So it will continuously divide. And another kind of assay or an experiment we do is we take a cell and inject into mice. There, if it forms a lump like a cancer, that means that cell is cancerous. Normally, if you put a normal cell into the mouse, nothing will happen. Okay, these are done in mice where the immune system has been knocked off. Okay, so these are the kind of experiments we do. So one thing is, in cancer cells, what are the strategies that have taken place? What are these defects? What has happened? The growth promoting signals have got activated, growth inhibitory signals have got disabled. So these are called, the growth promoting signals are called oncogenes, and the growth inhibiting signals are called tumor suppressor genes. Okay, so oncogenes are activated, tumor suppressor genes are inactivated. So these are the features that happen when a cell develops cancer. So how I describe that we have guardian systems. One guardian system is extremely important, one molecule actually. Any kind of stress or defect that a cell is undergoing, whether it's DNA damage, there is not enough nutrition, the cell has been occupied by a bacteria. So this cell is under stress, any kind of stress, when there is excess oxygen or there is insufficient metabolites. Okay, everything is perceived as stress, and all these kinds of stresses are gauged by a very important guardian molecule called P53. Okay, so this P53 is very important because what does it do? Once it sees that the cell is stressed, it sends signals to stop dividing, repair the damage, and if nothing can be done, it gives a signal for pressing the die signaling. So that cell gets eliminated. Most of the cancers, 90% of our cancers in humans are having defective P53 signaling. If any other molecule is defective also, if your P53 is normal, it is going to protect you. This is the reason why most of us do not develop cancer. Only a few people and a very small number of cells in our body become tumorigenic or cancerous. And of course, once P53 is not there, that will spread. Okay, please remember that a P53 is a very important, and we should be very grateful to nature that we have a molecule like P53. Okay, so there are some generalities with respect to cancer. Very few cancers are caused by inherited mutations. That means all cancers have undergone genetic changes. Okay, but where have these genetic changes come about? They've come about in the cells anywhere in your body most of the time. Very few changes have happened in your germ cells that have come from your parents. So inherited mutations are those that are coming from your parents. One example in the case of breast cancer is called BRCA. But if you inherit that, if there is only one copy, you will still not get cancer. If you are unlucky and the other good copy is also getting mutated, then that individual is likely to develop cancer. So if inherited mutations are very few, majority are linked to what are called somatic mutations. That means mutations in the rest of your body tissues. Those will not get transmitted to the next generation. So there's absolutely no worry that many of the cancers are genetically transmitted. A genetic change is there, but they are not transmitted to the next generation. Okay. And today we know that many cancers are linked to infections and also to environmental agents like smoking, tobacco chewing, and then also in recent times inhaled pollutants. And it can be occupational also. For example, people working in asbestos factories, their lungs get coated with asbestos dust, and those individuals are very prone to lung cancer. Okay. And then some things are from the dietary factors, like if you're not having enough fiber in your diet, then there is a risk of developing cancer in the intestines. Okay. And today it is also thought that obesity can enhance the risk of cancer burden. Okay. How much obesity contributes to cancer, we are not very sure, but definitely it contributes to the burden of cancer. Okay. So like I mentioned, infection, inflammation, and cancer. When you have an infection, very commonly, for example, cervical cancer in women is mostly caused by a virus. So if a virus has infected, it causes changes in the genetics of the cervical passage, and some of the cells become cancerous. I'll explain that a little. Similarly, there is a bacterium that can infect your stomach, that is called Helicobacter pylori. It causes ulcers, and also if that infection is there for a long period of time, it can cause stomach cancers. Okay, so infection causes cancer. And something called inflammation. Inflammation means where your immune system is producing agents like cytokines, is also known to cause cancer. So inflammation, an example of inflammation is something like in your joints. Okay, where you have joint pain and the tissue is inflamed, or in your gut. If you've heard of a condition called inflammatory bowel disease, where the digestive system, where the bowel is always having inflammation, where lot of immune reagents, immune chemicals are constantly secreted by a body, that causes tissue damage, and that can cause a trigger to cancer because the protective mechanisms are lost. Okay, I'll briefly mention how smart the viruses have been. We have understood all these molecular systems that control cell division, cell death only in the last maybe 30, 40 years. Okay, at the most maybe 40, 50 years. But the viruses, which have been existing for a very long time, know exactly which molecules they have to attack to survive in the cell, because virus cannot survive outside the body. So this HPV virus that causes cervical cancer, it makes two molecules. One is called E6 and one is called E7. What does E7 do? It increases, it triggers the signaling so that the cells proliferates a lot. But does our body like if it proliferates a lot? No. Our body will make, our cell will make no, activate P53, which will kill the cell. And then nobody will get cancer. But what does this virus do? It makes this other molecule called E6, which will go and attach to P53 and inactivate it. So it knows how to activate division and also prevent cell death, because if it inactivates P53, the cells will undergo continued proliferation, those cells will not die. So viruses act like this. Similarly, many other viruses which cause cancer like HIV, Epstein-Barr virus, HCV, Kaposi sarcoma virus, all of these viruses act in similar mechanisms by controlling the molecules within our cells. Okay. So these are some of the examples that cause some of the cancer causing viruses. I won't spend more time on that. Now, more recently, probably you are hearing in common parlance that stress, the psychosomatic stress, can cause cancer. Okay, this is a very difficult thing to scientifically evaluate. But today we know that people undergoing chronic stress. Chronic means continued stress. Somebody has lost a job and for years together they are not able to find a job. Somebody is taking care of a patient for years together, bedridden patients. That puts a lot of chronic stress. So in their case, what is happening? The chronic stress produces molecules called catecholamines or corticosteroids, which actually suppress our guardian molecules. So what happens? There is much scope that these cells develop this body which is under a lot of stress. This individual who is under a lot of stress develops cancer. But I won't say that we have total scientific evidence, because stress itself is a very difficult thing to define. What is stressful for me will not be stressful for another individual. So but there is biological mechanism known that if there is a stress, from psychosomatic stress, the brain produces chemicals which can suppress our immune system and enable forming of cancer. Now let us come and briefly talk about treatments for cancer. Earlier, what was happening? Any kind of cancer, we would just do some, it makes all, it kills all. Okay, so these were like, chemotherapeutic agents where all kinds of dividing cells would die, would be killed. That's why there were lots of side effects. The people would lose their hair, lose their teeth, then have problems in the digestive system. Okay, then other issues were, if it was a solid tumor, they could do surgery or give radiation. Okay, so but now these were causing too many side effects, and they were not specific. They were not specific to the tumor. They would act on all parts of the body. What do we want? We want the drug to act only on our cancer cells. So these older times, there was lots of problems. So we are now having modern modalities, because we have understood which molecules are defective in a particular individual. For example, somebody has leukemia, we know in that person what is the defective molecule. So we are able to now treat specifically to inactivate that molecule. So that is called targeted therapy or precision therapy. Okay, we can use immunological mechanisms or biological molecules which can go and act only at the site of the cancer. So this is very good. There will be very few side effects, but there are also issues like, very expensive. These, and we don't have this treatment for all kinds of cancers. For some cancers, we don't know which are the molecular changes that have caused this cancer. Okay, so at least we should remember that now there are specialized treatments which can, all these treatments which can act on various properties of the cancer. I won't go into more of the details, but we have these targeted therapies today. Okay, so I mentioned that tumors avoid the immune response. So we are now having mechanisms where we can enable our immune system to fight that particular tumor. Okay, I won't go into the names of these because they are called immune checkpoint blockades, but we have this capacity today. Okay, so then it becomes very specific. So we are coaxing our own immune system to fight the cancer, because cancer cells have learned, they are very smart, they avoid our immune system, and that's why they grow. So we can now use our own immune system to fight them out. Okay, but is it so simple? There are many hurdles. One thing is, when has the cancer been diagnosed? If it has been diagnosed at a very late stage, then it becomes a problem for treatment. Okay, so, and the other problem, major problem, has been how to handle metastasis, because the cancer would have moved to different parts of our body, and not all tissues are easily accessible. If a breast cancer cell has moved into the brain, we have a blood brain barrier which does not allow all kinds of medicines we have put into our body to enter the brain. So some of these problems we have to still overcome. Okay. And then the cause. Okay. And cost what? Not only of treatment, but the cost of disability of an individual. Okay, that person is not able to earn his living, contribute to the society. So the overall burden of cancer is presently very high in the society. So what is the means? Once you have cancer, you have to treat. But are there ways to prevent cancer? I mentioned that, some cancers are caused by viruses, by environmental agents. Can we now at least avoid those cancers? Because this virus or some of these pollutants, they are causing cancer because they are changing the genes in our body. Can we avoid them? Yes, to some extent. If we are aware of what these are, we can avoid them. So, and we can also make sure that we are doing our best for early diagnosis. Okay, so how can we get there? How can we get early diagnosis done? Through regular screening. Okay, so mass screening is done, for example, in the case of cervical cancer, something called pap smear. In the case of breast cancer. Okay. But in India it is not so popular, but it's a good thing that after certain age women go in for this kind of screening. And then also we can certainly avoid some of the risk factors like smoking, pollution, that is like, occupational pollution, because pollution from the traffic, all the city people, see people living in cities, I think it's very difficult. But one thing we have known is the masks have been very helpful in reducing the amount of pollutants that are going into our lungs. And there are also some of these items added to food for as preservatives, and some of the chemicals that are in the pollutants are acting as hormones and causing changes into our physiology. Okay, so we are finding that, girls are maturing faster. There is more infertility in the population. So these are some of the things that we are learning today and of concern for present day uh, human life. And how it will be in the future, it is very difficult. And also sedentary lifestyle and obesity is adding to all these problems. Okay, so some changes can also be made to the diet, that like eating fibrous diet, etc. Okay, now I'll just mention some of the common questions that many people have. If you are losing weight, is it cancer? Certainly not so. But if there is cancer in the body, they can lose weight. But every individual who is losing weight certainly does not have cancer. So now, is there an effect of cancer on the immune system? Or if our immunity is suppressed, can that individual develop cancer? Yes, where immunocompromised individuals are more at risk of developing cancer. Similarly, I mentioned stress. Today we know the mechanism, but there is no definitive scientific evidence that this individual's cancer has been caused by stress. That you can say, for example, due to smoking or due to this tobacco chewing. Those people develop oral cancers. Like that, we cannot say about stress, but there seems to be a link. Also, there is now, I will come to this aspect in the next slide. Now, body size and P53. See, for example, different animals, all of them are prone to cancer, but different animals have different sizes. For example, there is a mouse and there is an elephant. Both are mammals. Do you think that there is a difference in the number of cells, or the cells in an elephant are bigger? One would think that's not the case. The cells are not bigger in an elephant. They're the same size, but an elephant has many more cells than a mouse has. So that means the elephant's body cells have divided many, many more times. If the cells have divided many, many more times, how is it that elephants don't get cancer? Okay, so there has been a paradox. Do large animals get more cancer? No. that has not been the case. But we have understood that some large animals have specific protective mechanisms. And one protective mechanism, for example, we know in elephants is that they have many, many p53 genes. I told you p53 is a guardian molecule. So one mechanism of avoiding cancer is to have more guardian molecules. Like in us, there is only one p53 gene. Elephants have many, many p53 genes. Okay, so that is one protective mechanism. And another common question: do plants develop cancer? Plants do develop some benign tumors, but you cannot call them cancer because the systems are very different. The cells are very different between animals and plants. In plants, the cells do not move from one place to another, just as in the case there is no blood circulation, no, in the plants. And then plant growth is always in a restricted part of the plant, in the shoot of the plant. Okay, and plant cells have cell walls, so they cannot move. So really harmful cancer does not happen, just like in the case of animals, but they can develop growths. You must have seen some trees where the bark is having lumps of tissue. The bark is not smooth. So that is some kind of a benign growth in. So we cannot call that cancer. This is something that I told you about the paradox. If you look at the lifetime cancer risk, humans and dogs seem to have a high risk compared to whales and elephants. Okay, so this is the paradox. So they have some mechanism. Now scientists are trying to understand that mechanism so that can it help treat human cancers. But I should also mention before I end that there are several myths and misconceptions with regard to cancer. That means wrong ideas. Okay, see, these wrong ideas can lead to unnecessary worry and also prevent good treatment. Okay, so one thing we should all remember is cancer is not a death sentence. Today, the survival rate for many cancers is very high, and most people can lead a normal life. There are only very few cancers where we cannot intervene and not help much. Okay, there are a number of studies, now no studies actually, which show that there is something coming in the news that if you eat a lot of sugar you will develop cancer. No, there is no proper scientific evidence to say that. Okay, and people also ask this question: is cancer contagious? If somebody in my family is having cancer, will I also get cancer immediately because I'm staying in the same house? No, cancer is not a contagious disease. Okay, unless you have a viral transmission, like in the case of HIV. That how does HIV get transmitted? Body fluids. You have had a blood transfusion. Only in that case. Superficially, just like a bacterial infection or a viral infection, cancer cannot be passed on through the air or through touch. Okay, and some people feel that this person has a different attitude, that's why he's developed cancer. Certainly not. There is no link between development of cancer and the attitude of the person. And also some people worry that if there is a surgery, will it cause the cancer to spread? No, the risk of spreading to other parts of the body is very low. Because, and also there is in common press or common information that is transmitted in the local media, cell phones can be harmful. Today, as yet, we don't have any evidence that using cell phones can cause cancer. Okay, and so far, once a person has developed cancer, there are no herbal remedies that have actually totally cured a cancer. Okay, so keep that in mind. Cancer also goes through remission. Remission means sometimes it lies dormant and then gets activated. Okay, so remember that herbal treatments cannot necessarily cure your cancer. But okay, so one thing we should remember always is that several years of hard work that has come about through understanding cells and how our bodies develop has enabled us today to come together and make significant treatment modalities okay, to overcome many types of cancer. But we still have miles to go and conquer cancer fully, because cancer is a multifactorial disorder and not a single disorder. Okay, thank you one and all for a patient hearing. Dr. Jenny, I'm done. Any questions?
Yeah. మ్యామ్ ఆ సెకండ్ బురాన్ గారు క్వశ్చన్స్ వచ్చినాయా?
లేదమ్మా, ఏం రాలేవు.
బురాన్
Okay.
మ్యామ్ నో క్వశ్చన్స్ yet.
Okay.
Till now no questions.
Okay. So, should we wait a little longer or?
బురాన్ గారు, shall we wait?
అమ్మా చెప్పు.
వెయిట్ చేద్దామా? క్వశ్చన్స్ వస్తాయేమో.
క్వశ్చన్స్ గా ఓకే, ఒక్క నిమిషం.
No, that will depend on how many people have joined. You can find out probably, or if, because unless the question comes immediately, I don't know whether there are any doubts.
Till now we have no question, ma'am.
Okay. Okay, fine then. I hope the talk has been clear.
Okay, ma Jenny, conclude.
ఈరోజు జన విజ్ఞాన వేదిక మేడ్చల్ మల్కాజ్గిరి తరఫున కండక్ట్ చేసిన క్యాన్సర్ ఎందుకు ఎలా వస్తుంది అనే అంశం పైన అనేక విషయాలు తెలిపారు. అది ఎలా జరుగుతుంది అనేసి అనేక విషయాలు తెలిపిన రాధా గారికి ధన్యవాదాలు తెలియజేస్తున్నాము.
Thank you, madam.
థ్యాంక్ యూ మ్యామ్.
Can I leave now?